Welcome to H5Gizmo's visualizations of mouse embryos. This talk introduces the H5Gizmo's interactive visualization technology and describes how it has been used to build special-purpose tools to help analyze early mouse embryo development. My name is Aaron Waters, and I work at the Flatiron Institute in New York City. Before I run out of time, I would like to make sure I thank the people who've helped me with this work in many ways. In particular, Esther Postfall and Stas Schwartzman are the principal investigators leading the mouse embryo project. Lisa Brown, Maria Avdiva, Hayden Nunley, Abraham Corman, and Rebecca Kim Yip are researchers and scientists working on the project. And finally, I would like to thank Nick Carriera and Ian Fisk, my direct reports, who would provide me with the freedom to do stuff like this. I would also like to thank the ISMB conference organizers and referees for giving me the chance to give this presentation. Okay, what is an H5Gizmo? This busy slide summarizes the major points I would like you to remember from this talk. H5Gizmos allows a Python-based parent process, represented on the left in the diagram, to present a child user interface in a standard browser using HTML5 and JavaScript libraries, represented on the right in the diagram. The parent process and the child context communicate using standard web protocols, and they may run on separate machines. The parent process can use the extensive interfaces and scientific libraries available in the Python ecosystem, represented by the logos on the left. Example libraries include Scientific Python, scikit-learn, TensorFlow, PyTorch, and NVIDIA Rapids, among others. The Python parent process can also be used as a glue language to communicate with other environments such as MATLAB or remote services. The child, shown on the right, can use powerful visual and interactive capabilities available in the JavaScript HTML5 ecosystem, represented by logos and images on the right. Examples include D3, chart.js, cytoscape.js, highcharts, and leaflet, among others. These capabilities include high-performance graphics built using WebGL and WebCPU, for example, using the 3.js library. The major takeaway is that H5Gizmos connect a scientific workflow running anywhere on the internet to present an HTML-based user interface on any connected user-facing front end. About me and you. Let me mention a bit of background on where I'm coming from. I am a senior software engineer from the Flatiron Institute. I have varied experience in developing software solutions for startups, universities, large corporations, scientific projects, Wall Street, engineering firms, telecommunications, and government. The logos on the top half of the slide represent some of my clients and former employers. Presently, I'm developing scientific workflows and special-purpose interactive visual data explorers for scientific projects associated with the Flatiron Institute and its collaborators. About you. Who is the audience for this talk? This is a TechTrack, so I assume the audience is technical. I assume you have some programming experience and some familiarity with Python in scientific and machine learning applications, or you are familiar with similar platforms such as R or MATLAB. I also assume you have some knowledge of web technologies such as HTML5, CSS, and JavaScript. Let's discuss how the H5Gizmo's infrastructure has been useful for scientific research. Recently, I've been building H5Gizmo-based tools to help explore and analyze early embryonic development in mice for Esther Postfall and her laboratory in Princeton. This is an extremely cool and interesting project, and I'm delighted to be involved with it. Of course, what we would really like to know is how a single-cell zygote transforms itself into a human, but we have to settle for studying mice. Happily, at very early stages of embryonic development in mice is similar to embryonic development in many mammal species, including humans. The Postfall lab uses advanced microscopy to study the development of live mice embryos during early development, before the embryo embeds in the uterus. This project uses relatively new microscopy techniques and machine learning technology. The Gizmo tools I'm going to describe address the special requirements of these new methods. This slide illustrates how the post-fall lab observes mouse embryonic development using light sheet microscopy. Let me start the animation here. To view the developing embryo, the embryo is placed in a light sheet microscope, which is capable of extracting three-dimensional stacks of image layers at a moment in time. The microscope can be programmed to get image volumes for hundreds of timestamps over many days, and the procedure does not damage the embryo up to the time of implantation. In this slide on the top left, the embryo is extracted and placed in a transparent medium. On the bottom left, the embryo is embedded in the media is placed in the microscope, and the middle image shows the maximal value projections for the image volumes captured by the microscope and animated over the time steps. Each frame is a two-dimensional, I will start it again for the fun of it. Each frame is a two-dimensional projection of a three-dimensional image volume for a timestamp. The 3D volumes for the timestamp must be analyzed and classified, and that's where I come in. For example, the image on the lower right shows one of the special purpose tools for viewing image segmentation of an embryo image volume. Some of the basic questions for embryonic development are: how does the single-cell egg transform itself into many cells? When do the cells of the embryo, how do the cells move and divide over time? When do the cells of the embryo divide into daughters, and what are the fates of the daughter cells and their descendants? To explore these facets, we must determine which components of the image volumes for the timestamps correspond to the same physical cell of the embryo. We must also identify division events and mother-daughter relationships. The nuclear tracking tool, which I built for this project, allows an analyst to interactively identify and track cells of an embryo and their divisions in order to create a cell lineage tree that answers these questions. And now I'm going to briefly demonstrate some of the features of the tracker tool. This is the tracker tool. We can, let's switch to the microscope view. So here we have, on the left, we have a maximum intensity projection across all timestamps for the actual microscope data. And on the right, we have classifications of the cells into cells across timestamps. And we can move back and forth in timestamps to see how the cells move. Using the arrow keys, for example. And if there is an error, we can reclassify, say if this nucleus was incorrectly identified, we can reclassify it as one of the other nuclei. And we can also look at a specific slice. So this is what the microscope sees for a particular layer, not all layers together. And in addition to that, we can move up and down in the layers to make sure that we've identified the nuclei correctly. And we can also look at the mother-daughter relationships and the tree view of the cell divisions over time, and move around in time to see how things change. This tool also allows a researcher to examine a lineage tree created using automated methods, such as machine learning, and to assess where the automated method is correct or mistaken, and possibly correct the method if you want to move forward with the classification. It is also instructive to look deeper into the geometric structures of the embryo cell lineage to see the changes in size and shape of cell structures as they evolve and divide over time. The 3D lineage viewer allows a scientist to view, rotate, and zoom the geometry of cell structures while moving between timestamps. So let's start an example of the viewer and see how it works. So here I've created a script which starts a viewer for a given data set. I've simplified it a bit so that we don't have to load the data set. And we start that just as a standard Python script. And when the script starts, it says, oh, okay, it doesn't start, I've configured it not to start automatically. Instead, it prints a connection string. So if we open the connection string, it opens the visualization, and then we can load the data. And here we have a cell, a single cell that starting a lineage, and then over time it divides into 2 cells, and then it divides briefly into 3 cells at some point. 3 cells, there we go. You can see it moving, separating and dividing into 3 cells, and then 4 cells, and then we can rotate and take a look at how these cells are related. And in this case, we're looking at the cell boundaries, not the nuclei. The lineage 3D viewer provides a detailed geometric view of the cell lineage progression and allows the analyst to examine the geometric structure relationship between the cells and the embryo by rotating and zooming. The viewer does not start automatically, but prints a start URL, which we can direct to the browser or a choice. There are other start methods which open a new browser frame automatically. The lineage viewer requires a pre-processing step that extracts the isosurface geometries from the cell structures in the segmentation volumes. This preprocessor uses JavaScript and WebGL to extract the isosurfaces. So now I'm going to stop this interface. The cell lineage tracker, the first tool I showed, is actually a Jupyter widget and not built using H5Gizmos. The H5Gizmos methodology did not exist when I created the tracker. A Jupyter widget inserts a JavaScript visualizations and interactions into a Jupyter Notebook user interface. I was inspired to build H5Gizmos by successes and problems encountered developing Jupyter widgets. The Gizmo infrastructure addresses the following issues with Jupyter widgets. Widgets cannot appear in a new tab or window. They must live in an existing tab in a Jupyter interface. And the Jupyter framework is complex and can be confusing, and especially if all you want is only a simple graphical interface. H5Gizmos, by contrast, do not depend on the Jupyter infrastructure, and they can run outside the Jupyter user interface as standalone applications and in separate tabs. And this slide shows another example of a H5Gizmo which presents isosurface volumes of a 3D medical image, and it's running outside of Jupyter. How do you build gizmos? Let's look at a very short code example to give a flavor of the H5Gizmos implementation code. This slide shows an example demo called Hello 3 from the H5Gizmos package tutorials. This demo uses a button to update the current time display and modify other visual elements. This simple gizmo illustrates events, callbacks, and call-ins. Let's run it to see what it does. Again, it does not start automatically, but asks you to go to the link. We go to the link. It prints out a greeting and it presents a button, and then has another text area with no useful information in it. When we click the button, the text area updates to the current time and the header changes. And if we click the button again, we get the latest current time. And that's the simple demo. Let's talk about how it works. Line 5, 6, and 16 create two text displays and a button as the basic elements of the user interface. Line 19 combines the elements vertically into a single composite element. And line 30 starts the HTTP web server for the Gizmo and launches the task, which starts the Gizmo interface. This bootstrap boilerplate of starting the server and launching the task is similar for most Gizmo implementations. The click callback function defined at line 9 triggers when the user clicks the clicker button defined at line 16. This function calls in to the interface to modify the text displayed by the interface, displaying the current time. These interactions demonstrate that how the child context can alert the parent process to user interface events, and how the parent process can modify the child interface, possibly in response to an event. So gizmos have the following features. A gizmo is started from a Python-based parent process. For example, on the right, we see a gizmo started from within the Visual Studio Code editor at the top, and a browser-based Java child context connects to the parent process, in this case on the right in the browser tab underneath. And communication between the parent and the child is mediated by asynchronous web protocols. The child context can load and use vanilla JavaScript libraries and other web resources, and the user interface may be constructed by combining basic components like buttons and images into configurations using composites like stacks, shelves, or templates. In practice, a program that uses HTTP outside of protected environments should use secure communication methods and well-known connection ports. The H5Gizmos package provides a proxy server implementation to facilitate remote connections between a parent process and a child context. The proxy server is automatically installed as a Jupyter Server plugin and works with the Jupyter Binder or Jupyter Hub framework. It allows the child to use standard URL entry points to connect to a gizmo, to gizmos running behind firewalls on remote servers. This also works from within containers inside, for example, Kubernetes or Docker Swarm, running in environments such as AWS or Google Cloud. Summary. This slide summarizes the talk. The animation shows a gizmo that explores the behavior of a Lorenz attractor system under different configurations, built using numeric Python and Matplotlib with jQuery UI sliders. A gizmo implementation connects a persistent server-side Python parent process to a client-side child context based in JavaScript. The Python parent process provides access to powerful scientific and machine learning platforms and tools such as numeric Python, scikit-learn, PyTorch, TensorFlow, NVIDIA Rapids, Matplotlib, and Seaborn, among others. And the Python process can also act as a glue language to access external systems such as MATLAB, R, Stan, or remote REST APIs. The JavaScript child context supports extensive and diverse interactive user interface components, including D3, jQuery, Chart.js, Plotly, as well as GPU-accelerated graphics using WebGL and WebCL with libraries such as 3JS or Babylon.js. Using a proxy server as a connector, a Gizmo implementation can present an interactive graphical interface for a remote process anywhere on the internet on any connected user-facing device that supports standard web technology. Please download the open source packages described here from GitHub and give them a try. Thank you for your attention.